I'm very happy that we start our third Philosophy of Cancer Biology workshop. And our first speaker is Pierre Soubeyran. Pierre is at UniCancer, Bordeaux. He's part of many very important organizations here in Bordeaux. And we are very, very happy to have a clinician who is also someone who likes thinking about cancer research in general and the clinical dimensions of cancer more specifically. So we're very happy and honored that, Pierre, you accepted to give a talk today. If this is okay for you, Pierre, I think you can share your screen if you have something to share with us, and that would be wonderful. Thank you so much, Pierre, for accepting the invitation.
So thank you to give me the opportunity to talk to you about the management of vulnerable and frail patients with cancer. I think that's a very hot topic, and I will try to give you a clinical point of view, and then we will continue, because I think also it's the right time to discuss about biology, aging, and cancer, where we have a lot to do to add to the clinical approach. So I will present you an application that we had to H 2020. We will have the answer within the month. I'm eager to have the answer for sure. And that's about the clinical trial to try to improve the management of all the patients with multimorbidity. I will get back to this. So what is the current situation? Well, the current situation is that the population is aging, and it's all over the world. And it mainly concerns the non-developed country right now, the emerging countries. Well, they have a figure here to show that's what happened in the Nouvelle-Aquitaine. That's just an example, but you have the situation in 2013 and 2050, and you see that there is one population that will increase, that the patients who are older than 65. And that will be 650,000 new older patients that will come in Nouvelle-Aquitaine within these next 30 years. The second point is that the cancer is a disease of older patients. If here you have a presentation of the incidence of cancer according to age, well, that's not a new fact. It's something that is very strong, that shows that there is a strong increase of cancer incidence with age. And the last point is that age is clearly related to survival of patients with cancer. And you see that in all types of cancer, survival is decreasing with age. That's important disparities. So why? That's an important point. Why do we, how can we explain these problems? Well, first explanation that is, well, very simple, is that with age, there is an accumulation of comorbidities. You see on this slide, so it's the proportion of patients with comorbidities, the number of comorbidities, and the age. You see that above 50 to 60 years, the increasing is really sharp, and it can goes up to more than 90% of patients with at least one morbidity above 80. That's really important, and you even have close to 50% of the patients with well, at least three comorbidities together at the same time, which makes the situation complex. That's the first point. Then a second point, that, well, these older patients are exposed to a high risk of unplanned hospitalization. That's a first series of 25,000 patients from the California Cancer Registry. Well, whatever age, so no relation to age right there, that what you see is that the proportion of patients that will experience at least one unplanned hospitalization is quite high. It's highly dependent on cancer. You see, it's lower in prostate and breast cancer than in colorectal and pancreatic cancer for various reasons. It may be quite frequent, that is 3 or more unplanned hospitalization during the first year of the beginning of treatment. Again, there is a clear relationship with the type of cancer, but it may be 1 patient out of 5 that may experience this problem. Then if we look at now at the theories of older patients, older than 65, 30,000 patients, again from the USA, that's the Texas Cancer Registry, you see that first, these events, these unplanned hospitalization are predictable. Well, in this area, they looked to the factors that predicted, and you see that socioeconomic problems are major, advanced disease, high Charlson Comorbidity Index. I don't know whether you know Charlson Comorbidity Index, but that's an index of comorbidities that group only severe comorbidities with an influence on survival. So a high Charlson Comorbidity Index predicts also the unplanned hospitalization. That's the multimorbid patient I was talking about. And various types of cancer. This series was limited to the GI tract cancer, so gastrointestinal cancer, though these are the severe one, esophagus, gastric, and pancreatic cancer. Well, the two last ones are more specific to the US population with Black race and eligibility to Medicare and Medicaid. But just remember the four first one. It's illustrated there. You see the patient with at least 3 comorbidities according to the Charlson Comorbidity Index have a risk of being hospitalized that is the highest. And last point, well, most of these, the most frequent complications that lead to unplanned hospitalization are not related to cancer. Well, probably indirectly, but they're not related to cancer, which means that we have the capacity to intervene in it. That's the fluid electrolyte disorders, intestinal obstruction, pneumonia, congestive heart failures, and so on. So the reasons that lead the patients to hospitalization in an unplanned fashion are predictable if we work on that, and they may be also preventable. That's the role of the geriatrician. So I'll get back to the kind of way of thinking, why these things happen, try to explain. Well, I think there are 2 points I have to tell you as a clinician. The first one is, when you have to deal with the management of an older patient with cancer, you have to think about why you treat them. What is the endpoint? What is the objective of the treatment? And for this, there is a balance between 2 endpoints. That's disease control. We should control the disease for sure. That's always the objective, but without having not too much trouble, not too much toxicity. And for these endpoints, we are quite comfortable because we have tools to predict and quantify these 2 endpoints. And according to this, we will decide whether we should push up to cure in fit patients or whether we should just try to keep disease control, just to keep the patients independent, which is a major endpoint in all the patients, to keep them independent. Well, but there's another balance we have to take into account. That's a balance between quantity and quality of life. Indeed, quality of life is decreasing with age. That's quite obvious. And consequently, the quality of life is getting more and more important for these patients. And in a way that you cannot predict. You cannot, as a physician, tell whether the quality of life is more or less important for these patients. Well, we may have opinions, but indeed, this point is highly dependent on patient views, and you cannot predict it. You have to ask the patient. That's something major. So that's the first point. You have these 2 balances to manage. The second point is, well, when you see a patient in your waiting room before the consultation, well, on first sight, you see 2 groups, the independent and the dependent patient. Well, the patient who have problem to raise from his chair and to walk to your room probably is a dependent patient, the one that is in a wheelchair for sure too. But the reality is not that. We have at least 3 groups. Well, it's just theoretical because the reality is just continuous. But indeed, it's important to think in this way because, well, you have the frail patients, those you see them in a wheelchair, difficult difficult to walk and so on. These are unable to adapt to stress. That's the lower curve. So when they have an illness, their dependence will be impacted, and well, they will, with attention, they will recover, but they will always recover partly. So they will get to a lower level of dependence. On the other hand, you have the fit patients. They are able to adapt to stress as younger patients, no problem. And in between, you have those patients that look fit, they are independent, well, they have no particular difficulty, but, well, they will need help to recover. Otherwise, they may get into dependence and may not recover. So you have to identify this patient, and it's not possible or quite difficult to identify them only with the eyes of the clinicians. You need some tools, and this is what is called geriatric assessment. So what can we change to this situation? Well, the geriatricians know about that. Since many years, you see, this paper is from Lawrence Rubinstein, is 1984, that many years ago. And they work in the general population, where it's clearly demonstrated that geriatric management improve survival. You have here the percentage of patients, the mortality indeed at 1 year. It's geriatric evaluation unit, and then control. You see that you decrease the rate of death in 1 year by a factor 2. Second point, the number of patients at home at 1 year is 1.5 more if you manage them in a geriatric evaluation unit. And finally, which is not expected, well, by preventing complications, but by maintaining patients at home, there are cost savings. And really that's important, because one main reason that make that the physician don't want to perform geriatric assessment and management is because they think it is costly. But at the end, well, this is an investment at the beginning of the management of the patients, so that at the end there is some cost saving for the society. So that's demonstrated in the general population, but that's not demonstrated in cancer patients, because cancer is a tough comorbidity that may kill patients. So the impact of cancer may change, may make this benefit disappear. That's the reason why we have launched a clinical trial that is currently ongoing, that the PREPARE phase 3 randomized trial. So we randomize patients that are supposed to be vulnerable or frail, thanks to a tool that we have developed, which is named the G8. It's a questionnaire of 8 questions that try to find some vulnerabilities in the patient in terms of mobility, in terms of cognitive problems or mood problems, in terms of nutrition, and so on. Then if the patients are positive, according to this G8 questionnaire, they are randomized between standard management, that's current oncology management, or to case management, where beside classical oncology management, you will have some case management with a nurse and a geriatrician that will make an evaluation at the beginning of the treatment and propose an intervention plan for a year. Well, the nurse will be on first level to support the patient and have him on phone or in clinical consultation once a month for the first 6 months, that is during the treatment. So it's ongoing. We have 400 patients included up to now. We need more for sure, and we expect the results within a few years. Well, clinical research is always very long. You have to understand that. Well, the first step is just to convince colleagues and raise money, and the last step is also quite long to recruit patients and make the analysis. Well, what do we do perform practically? Well, we perform a geriatric consultation at the beginning with full geriatric evaluation. It's performed with the nurse. Then the nurse is working with the patient with phone follow-up or clinical consultation. The nurse will call for the help of the geriatrician as much as necessary, and at least at 3 months, 6 months, and at the end of the 1-year period of follow-up. The objective of this trial is to improve a combination of survival and quality of life. I was talking to you about endpoints. Well, to have patients have a longer survival or better disease control is certainly a good thing. But if it's to live in a retirement home, not living at home, or in poor quality of life, that doesn't mean anything. So we have to take into account some patient-related outcomes, such as quality of life. And that makes the trial more complicated because we have to collect quality of life all along the management of the patients. But that is just mandatory if we want to treat all the patients with cancer. So we'll have to wait. So, but is it sufficient? That's the question. Well, indeed, the situation currently is a bit complicated. That, well, trying to model, I'm not sure that's the term, what is the current disease-centered management, where you see on the left side the hospital and the right side the home-based network that take care of the patient with the neural practitioner, a home-based nurse, some morbidity specialist. And on the hospital you have the oncologist, maybe the geriatrician in some case, other morbidity specialist, and well, all these health professionals have to communicate together to get the information and make the right decisions. Here we put what are the decision centers in the dark blue circles. Well, the oncologist, when we are dealing with cancer for sure, the GP for sure, also in many times at the moment of cancer management, the oncologist is the main decision center. And for the other morbidities and the morbidity specialist that may be in the hospital or close to home. Well, what we add with the prepared trial is that we put the geriatrician in the decision centers together with the oncologist, and we want to demonstrate that the geriatrician will improve survival and quality of life. Okay, that's one step, but we won't change the whole thing. Well, the geriatrician will serve as an integrator of all this knowledge about the patient, but the way we work with the geriatrician doesn't change that much. So we want to improve over this, and especially for a proportion of this patient, which is quite frequent, that the patient with multimorbidity. What does it mean, multimorbidity? It means that you have at least 2 morbidities together, though that will be cancer and some kind of severe disease like cardiac disease or any kind of disease that may impact survival. So we propose, and that's the H2020 application, to change to a patient-centered management, where the advanced practice nurse, which is currently developed in France quite lately as compared to Anglo-Saxon countries, well, the advanced practice nurse will be the main contact for the patient. And we want not to have only the oncologist, the geriatrician, and the GP do their job. We want all the health professionals that take care of the patient to be linked together to take decisions. So we want the patient to be managed not on a disease-centered approach, but on a patient-centered approach by a health professional consortium. Well, that's the main point, that's the main difficulty, because it's organizational. So it will be quite difficult and certainly not possible to change everything at the same time at the first moment, but just to focus on multimorbid patients, who are the main complex ones. For this, we will use some web-based meeting to help these people work together. It has to be organized, taking into account the daily planning of all these people. So that will be tough for sure. But to make this feasible, we need the digital tools. So first, we need to structure the data that are necessary for the decision at the moment patient has cancer and they have a morbidity. So we have devised with a startup named MyPol, a way to structure the clinical data that come from the e-health records to put on the screen clearly and to ensure that we have all the necessary data. Just to be more clear, the objective is to have certainly the cancer data, but also the geriatric data, the comorbidity data. And it's not only to know that the patient has diabetes, but that he has diabetes with 1 or 2 medications. So, and how is the diabetes equilibrated and so on and so on. So we put all this on the screen so that you will see the holes, what things are missing. So that's a kind of training tool for the physicians. Then we need also the home-based patient-reported data. So we'll ask the patient to give information on a weekly basis about what happened when he is at home, so that we may take decision on a real-time fashion. And then we will work on the patient. Well, for these activities where the patient is independent, he has the capacity to train himself. So that's what we call self-management. Then we will work with patients. For this, I said from the beginning, quality of life is only available through patients. To develop these kinds of tools, we need a co-design with patients. So we have organized things to have the possibility to co-design with patients through focus groups and so on. And then we will have to collect patient and expectation and put all these in some databases. So final point, how to make the demonstration. Well, we will use a stepwise design. So to explain briefly, that's not a randomized phase 3 trial where you randomize patients, you randomize the hospitals. So if you look at time according to the centers that we include, at the first step, that is 2 months in our case, every center will perform as usual and we will record the information. And by random, at the second step, 1 center is coming in, the innovation, and the third step, a second center, and so on. It's always by random. We will include investigating centers from referral centers, but minority, a lot of community hospitals will be included because that's where the older patients, the older frail patients are managed most of the time. We will perform 2 trials because the health system is very important. The health system should have the capacity to use this kind of approach, which is complicated. We decided to perform 1 trial in France and 1 trial in Belgium and Netherlands. There will be 8 centers per trial and over 650 patients per trial to make the demonstration. Well, I will go rapidly on this slide. Well, we propose a new model to improve the quality of life of patients. That's the endpoint of the trials. We will work on the cost efficiency because we have to show some cost savings, although we imply a lot of people there. We will work on data management because I think that's the next step, maybe to work with artificial intelligence, machine learning, and so on. So the collection of quality data will be very important and allowed by this new approach. We will develop an ICT tool that will be important for this patient. It's information and communication technologies. We will work on the social and economic relevance in the various European countries and draw recommendation for the replication in different European health system. So as a conclusion, well, the management of all the patients all the vulnerable patients are certainly to change. We have a lot to do, and we have the capacity to do, but we need to launch some organizational changes. Sorry. We need digital tools, and we have to implement this in our hospitals, which is beginning already. We have to include patients in the design of the clinical pathways. That's something that is really very important, I think. And well, we have to work, and this proposal is a one step towards some shared decision making. So thank you.
Thank you very much, Pierre. Okay, I have a question just to start the discussion. So there is a very obvious potential contradiction, so to speak. I'm sure you're aware of that, which is that I really love this idea of really doing things with patients, and this idea of co-designing with patients, I think is very interesting. One thing that strikes me is that the patients who are the most frail, the most in difficulty, I guess it's very difficult. I mean, it's more difficult than for other patients to talk with them, to know what they want, to involve them in the decision. So isn't there a risk that we take more into account the opinion of people who are not so fragile, not so in a bad condition, and to the maybe at the expense of the most frail ones?
That's a real difficulty. Well, to work with patients where we need to have the real patient to discuss with us. Mm-hmm. That's exactly what you say. And well, you observe already that there are no older cancer patient association existing, because well, already to have a cancer when you're old with a lower life expectancy, with poor chance of survival in some cases, like that. If you get into an association, you will have people that may die quite rapidly, especially the frail patients. So indeed, well, there is no solution for that. The solution will only be in the discussion between the physician and his patients. That's where certainly the long-term relationship that we have with patients with cancer is something very important. That's also why the GP is getting more and more important, because he has a long-term relationship with the patient. So on the daily basis, it's manageable because we have this long-term relationship. And if the oncologist managing with the GP and all the other specialists, is manageable. Mm-hmm. Okay. Well, that's a tough question when we are talking about co-designing trials and studies with for these older patients. And the only approach we can have is to deal with, on one side, with cancer patients who are not old, not frail, or older patient but fit, on to work also with citizens, not work with people from from cancer only, but also citizens that have already experienced. That's I would say it's palliative. That's we have no choice in this way. Yet I think we have to work on that. You're right, and we have to devise new approaches to understand what is feasible. And one thing that we have to collect, I was talking about shared decision making, is to ask the patients before treatment what he expects from this treatment. Because, well, you think that the patient wants to be cured from his cancer, not to have too much toxicity when he is old. But indeed, that's not the case for all the older patients. It's more the case than you expect, obviously. But indeed, many patients, they want to keep autonomous. That's the main objective. So the first thing is to ask patient expectation, to have this information at the time of the treatment decision making, so that we take the good decisions. That's the approach.
Yeah, very interesting. Thank you very much. We have a question by Sarah Green.
Okay, thank you. I'll try to be quick, and I apologize for not having the camera on. I'm working on it. You mentioned that you'll have these web-based meetings where you bring people together with different backgrounds and specialties. What do you see as the main challenge? You mentioned this will be tough, but is it the toughness of this, is that due to different specializations, or is it more practical?
Well, I think it's practical. The first thing is practical is, well, when you perform a multidisciplinary tumor board centered on the oncologist, well, the oncologist just have to work together to define a moment within the week in the agenda to group together. When you deal with specialists from who are home-based or from other hospitals, well, because this consortium that I want to group of the patients is one of the patients is not one for every patient. It's the physicians of the patient that take care of the patients on the daily basis. So, well, for each patient, we will have to find the best moment to have these discussions. That's where the role of the advanced practice nurse is really central. I think it's feasible, but that's something that should be organized. They should have, for sure, first, the same set of data. That's we will work on that. And you have to share exactly the same data, but then we will have to find a moment for this discussion. And I think that's the most difficult, because everyone should modify their organization. That's the reason why a trial is a nice approach most of the time, because especially for just a short moment, you have to concentrate and try to find solutions. And there you see whether it's feasible. What makes it possible is that you save a lot of time if you have everyone around the table to take the decision, instead of having many calls to reach one or the other who won't be available when you are available. So I think it's, on the long term, it's certainly very feasible. But on the short term, we will have to convince physicians to accept this organization.
Thank you very much, Pierre. Thank you, Sarah. And now it's time to listen to Laurent Cognet. Laurent Cognet is another of our local stars here in Bordeaux. And Laurent does something which we think is extremely interesting. You saw a little bit of that last January, if you were with us, with Pierre Massois, for example. There is a big and very interesting group of people here in Bordeaux using different sciences and technologies to shed light on cancer. So in fact, Laurent is the PI of a group specialized in nanoscience, biophotonics, and super resolution microscopy, and they use their impressive knowledge to shed light on cancer. So Laurent, this is a real pleasure for us to have you today, and now the floor is yours.
Thank you, Thomas, and I'm very happy to be here. So I try to share my screen. So do you see it?
Yes, it's perfect.
It's working. Okay. So thanks, Thomas, and I'm very happy to be here. But I have to say that I kind of struggled at first to know exactly what I would present. As you will see, we have not yet performed many results in the field of cancer, and what then I decided to show you is kind of our roadmap that we'll pursue to try to help the field of cancer. And as you can see here, my title will be dealing with so these new tools and to focus on the tumor microenvironment. So before I forget, and I would like first to, oops, so that doesn't work. So why? Okay, so behind what I will present you, and there are faces, there are people, and you will see a few experimental results, and that's the people from the group that participated to the work I will be presenting, and also without forgetting some of our former members that were important. I would show you and take some example at what we perform in the neuroscience, and we had some very nice collaborations with people in neuroscience, Laurent Cognet and Erwan Bézard in Bordeaux. And so the roadmap that I will show you has been built also with Pierre Massois and Frédéric Saltez here in Bordeaux. So let me just present a very simple question that that actually is very central to my research at the moment, which is to try to understand how do molecules actually navigate in a biological tissue, in a live biological tissue. And if I try to translate that to the cancer field, I would like to try to understand what's going on inside the tumor. And of course, why should we care about this and why is it important? But the first thing is that in the so-called extracellular space or intercellular space, actually you have a lot of molecules which are there for signaling. You have in disease, of course, you have some signals that will help or promote disease spreading. You have everything about food and energy supplies. Also, the tumor or the tissue might create some waste, toxins that need to be removed or not. And obviously, this space, the way the space in between cells is very important for immunity. That's where you have antibodies that will navigate. And also, if you want to create some drug deliveries or immunotherapies, maybe you have to understand how these molecules are able to navigate into the tumor. And now, if I zoom on really what is the microenvironment of a tumor, a lot of people look at what's going on in the blood-through-driven transport, how molecules go from the blood to the tissue, the transvascular transport and these things. But you have to understand that this is not the end of the story. And actually we're passing from this space to the tumor side. There have been some calculation and some evidences showing that actually you lose 3 orders of magnitudes of molecules that that are able to go into the tumor. So that's a little bit to motivate what we are trying to do and what I would like to address in the coming years. So actually there are 2, and that's what I will present you, our approach to do that. Maybe it's a very oversimplified approach, but sometimes, you know, coming from another discipline to tackle a question like this can be very useful. And so our roadmap would be 2 things. The first one would be to try to understand how does it look like? How does the tumor intercellular space look like? And the second thing, of course, is once we know how it looks like, what are the rules that dictates how different types of molecule can navigate in such spaces. So if you want to illustrate that, and I hope I'm not too simplistic here, if you look at different tissues that you can find in cancer, seems that structures are so different, and so that there is not, of course, there will not be a uniform answer to that, but it seems that there are some specialized organizations. But so I'm not able, of course, to analyze these things today. And as a physicist, I would rephrase this way. So the question I'm addressing here is, does the intercellular space look like the space in between roads, or is it like you know, sheets, and that you can navigate in planes, or is it more like sponges? Or maybe we can see cells like balls, and you have some space like you would have in a messy thing like this. Or maybe it's not organized sticks like this, but it's totally inorganized, like you would see maybe in polymers. So that's it. So today, that's my vision of tumors on tissue. Okay, that's this kind of things. That's what I will look around. Now, the second thing to look at is what are the dimensions we're talking about. So let me take the example of healthy brain tissue here. An electron microscopy over the years gave us some hints about that. And I have to say that we had a very bad view for many years before people were able to do cryo-EM. And people realized actually the space was much more diverse and much more larger in between cells. And that's what you have here in blue. That's what you would do if you would do a chemical fixation before the cryo-EM. before doing TMM images. So actually the dimension that that we think are correct today are in tens of hundreds of nanometers. So it's very small, but it's not vanishingly small as you would see for a tissue like this where everything has collapsed. So it's a space which is quite important, and we think that it's important to study it. So now the question is, okay, obviously this is not a tissue that is live and that you will not look at molecules moving in it. So we have to be able to visualize this dimension in a live tissue and look at the movement of molecules. And so you know, of course, that if you want to look at an organ of a tissue, for any scale that you want to look at, there are different strategies and different investigation strategies. And in our case, what we want is nanometric resolution, as I just said, in a live tissue. And again, nanometric resolution is provided today by electron microscopy, but it's not applicable to live tissue today. So what I will present you, and that will be somehow the way my presentation will follow, is that we'll do optical microscopy, because optical microscopy allows you to do live tissue imaging. But if you want to have the resolution, we have to introduce new strategies, and that's something that we have developed over the years, single molecule optical microscopy. And to apply it really to thick tissues, we'll have to think at novel single molecule probes, and that's again something that I will talk about. So what about, okay, optical microscopy and resolution? What can we do? So there is a basic limit in, I'm sorry, that will be the only equation of my presentation, which is the equation which tells you about the resolution that you can have with an optical microscope. And what does it tell you? That it depends on the wavelengths of the light, because you use light to probe your sample. And it will also depend on what is called the numerical aperture of the objective, the way you collect the light. So let's think about a simple structure, which is 2 points. You want to see, are we able to distinguish 2 points with an optical microscope? So if those 2 points are too close to each other, actually, if you don't resolve it, that's what you get. So it means that you don't have enough resolution. And the limit, actually, in optical microscopy with this formula is around 300 nanometers. So it means that when you have 2 points which are not separated enough, you will not resolve them. And in general, if you apply that, so people like to compare actually the scale of things, so different somehow, okay, biological things, if you will, with the wavelengths that we are using. And actually we are using here visible light, okay, that's where we are here. And it means that if we cannot resolve things that are smaller than the wavelengths of the light here, actually everything which is below, let's say 100 to a few hundred nanometers, you will not be able to resolve them. And that's exactly what we want to do. So we have to find find a solution to fight this. Okay, if I rephrase that to make things a bit more visible, if you take this image, electron microscopy that I showed you, and you apply actually the limit in resolution that we have, that's the kind of image that you would get. And obviously we are not able here to resolve what we want to do. But there is another thing which is important to bear in mind, is that tissues, when we think about intact tissue, they are not transparent. And when you work at visible light, actually, well, we see each other, we are not transparent, the light doesn't go through us. But maybe you have done this very simple experiment where you put light through your fingers. So that's one of my daughter's hand here. And you put light and you see that red photons actually penetrate a bit more in the tissue than actually blue or green photons, which don't go through. And actually the reality is that it's even better in the near infrared. So if you want to be able to perform imaging through a tissue with light, you'd rather go here in the around 1 micron in the really near infrared. So that's something to keep in mind. So to summarize, our approach is based on the fact that we want molecular sensitivity. We want to see molecules moving in the tissue. We want molecular resolution. We want nanometers in the resolution. And we want to work in live cell and tissue. And one day we would like to go in vivo. So what is the solution for resolution? Actually, so our solution will be based on single molecule detection, single molecule fluorescence microscopy. Imagine here that we are with a microscope. So these are real images that we perform with a microscope, but instead of having many molecules, we have only a few, and these molecules are well separated from each other. And all the molecules look the same with a resolution given by the microscope. It's something like 300 nanometers. That's what you can see here. But now there is a trick. The trick is, okay, let's me zoom on this one. Okay, 300, 250 nanometers was basically the same. Okay, maybe I cannot resolve this molecule. But what I can do is if I know that there is only one molecule here, I can find its position very precisely. And the position is simply given by the center. If there would be two molecules here, I would not be able to resolve them. But when I have only one molecule, I can say, okay, it's here. And so that is what is called the super localization. We can find the position of a molecule with very high resolution, high accuracy. And so this is something now that we'll use. So let me explain you how we'll use that for our problem. So our strategy again will be to measure dimension of the intracellular space in biological tissues and molecule diffusion. So now imagine, so it's a Gedanken experiment that we are able to insert a molecule in a tissue, in the intercellular space of a tissue, and that this molecule is moving. Imagine that we are able to detect it, okay? The molecule is moving. That will be the image that we'll get. But now I told you that when you have only one molecule, one at a time, we can find the position of this molecule. So it will be here, this point, for instance. And imagine that we are able to do that while the molecule is moving. So at each position, we'll be able to find where the molecule was. And now imagine that the molecule is diffusing, but at the same time it is exploring its environment very extensively. So we have many data points here, okay? So we'll be able to recreate the dimension of the extracellular space, okay? And at the same time, of course, because the molecule was moving, we'll be able to study the diffusion properties of the molecule because we'll look at how fast it goes in here, okay? The second thing to do that, of course, I told you just before, is that we need to do that in a live tissue. It means that we need to do that in a thick environment. And we'll need to have molecules, probes that are emitting in what is called the optical window in the near-infrared that I told you before. And we had to develop that in those last year. So now let's see what kind of molecules we'll need to do that. So imagine that you take a spherical, very small spherical particle or molecule of particle. So the tissue that I will talking about first is agarose gel. So it's very easy tissue to analyze. And if you think that you use 20 nanometers beads, which are fluorescent, that's what you would get. Actually, it's exactly the situation here, except that the molecule goes very fast everywhere, and you are not able to have a lot of points to make your trajectory. And what we discovered those last year, actually, that we have to go to very unconventional nanoprobes, which are carbon nanotubes. So carbon nanotubes are from the family of carbon-based particles. So you have diamonds, graphites that you know very well, fluorines that are small balls. Nanotubes are those ones. It's 1D objects. And then maybe you heard about graphene, which are really flat samples like this. And the good thing with nanotubes, so nanotubes are 1 nanometer in diameter and can be pretty long in length. And the good thing is that they emit light exactly here. They don't photobleach, and they are very small and at the same time very long. And that's something which seems to be contradictory, but which is very important because look, here you have a nanotube that we place in agarose gel, the same than this one. And you see that the nanotube is able to move inside the agarose with like a snake. And we can look at this guy for hours. Actually, there is no photobleaching. And again, it's one object. Huh? It's only one object. Okay, but here maybe it's not exactly what we want. We would like something that moves a bit faster. So if you change the length of the nanotube, you can be in this regime here, the bottom right video, where you see that the nanotubes is able to probe its environment very efficiently. And again, now we are exactly in this situation. We have an object that is able to be tracked for a long time in the near infrared and to provide such images like this. So our demonstration, so we did that in, so again in neuroscience, I told you at the beginning, we injected some nanotubes in the brain of rats and rodents in general, also in mice. We then performed, we created brain slices that we put, live brain slices that we put on our microscope. And this is what we get. Actually, you see that we are able to track the movement of carbon nanotubes inside brains like this. And by doing the strategy that I told you before, actually we can pinpoint the position of the nanotubes as a function of time. And with very high resolution, now some kind of paint the intercellular space like this. So we create maps like this. And the resolution that we have now is 50 nanometers. And that's exactly what you can create. It's mapped like this. And these maps, actually, you can compare them to what we find with electron microscopy and compare with chemical fix. And if I rescale this image that I showed you before to the one we observed, you see that we exactly find the same dimension that people had found with electron microscopy, but now we can do that in live tissue. And so that's really the novelty. And what we have on top of that, of course, is that we can analyze the diffusion of molecules in all these tiny regions, and to understand if a molecule is able to go fast, go slowly, stop and go. And so we are exactly where we wanted. So that was in brain tissues. And we even did that in Parkinson brains. So I will not go in the details of all this work. Very recent work performed with Erwan Bézard at IMN in Bordeaux. But we could see that the diffusivity actually of the molecules is very different, while the dimensions are only slightly different. So these are the dimensions. So in a healthy brain, here in the Parkinson brain, so it's a model again, it's in mice. And you see that the diffusivity is also very different. So today, I didn't want to go really in the biology of that. And it's just to show you now, we have the tools to see very subtle changes in dimensions. See, the dimension is only very subtly by 100 nanometers changing in the tissue. It changes dramatically the diffusion behavior of the molecules. So my last slide will be this one. And now, of course, our roadmap, again, we have not yet results in tumors, but we are working on it at the moment. So our roadmap now is to revisit the diffusion properties of molecules, of nanoparticles, or drugs, or whatever, what you think that might play a role in a tumor, and to stop to think that things are very simple. It's just when you have a molecule in the tissue, it always behaves the same. And it's really underseen today. We even don't know, of course, you know better than I do, that people talk about lymphatic drainage. We don't know if there are some fluxes in tumors. This has not been seen today, okay? We don't know if an antibody can go really inside the tumor without injecting it right away in the tumor. So we believe that this will be extremely important to understand the architecture and the different properties of molecules. And I didn't have time also to talk about another project, which is also based on carbon nanotubes, but I prefer to make a choice that we are doing also with the group of Fred Saltet, is to create a multiplexed markers for analyzing biopsy, and these are based again on carbon nanotubes. We play a little bit with the carbon nanotubes to create new families of nanoprobes for immunoanalysis of the tumors. So I will finish with this picture here that I like to present, and just to tell you that sometimes to go from point A to point B, it's not the straight line, which is the best one to go. And to go to other disciplines and visit other landscapes is sometimes very interesting to try to address a new question. And I hope it's not a too simple question that we'll be able to have answers soon. So I think my time has gone, and I will thank you for listening to me.
Thank you very much, Laurent. That was great, really interesting, and I think a perfect example of what physicists and physics in general can bring to biology, as you showed with neuroscience, and also to oncology soon. So that was really super stimulating.